Hello everyone, welcome to this explanation of the basics of the autonomic nervous system. In this presentation, we are going to be discussing, first of all, what is the autonomic nervous system, what are the autonomic nervous system divisions, then we are going to move to the effects of the autonomic nervous system stimulation. We are going to describe the basic anatomical differences between the sympathetic and parasympathetic nervous system divisions. Then we are going to be talking about the acetylcholine receptors and about the adrenergic receptors, so we can understand the main differences between the different branches of the autonomic nervous system and understand how the central nervous system can take control of changes in the internal environment of our body. What is the autonomic nervous system? The autonomic nervous system is the portion of the nervous system that innervates smooth muscle and glands, influencing the function of internal organs that regulate the heart rate, arterial blood pressure, digestion, micturition, defecation, sweating, and sexual function. What are the two divisions of the nervous system? The autonomic nervous system is subdivided into two separate nervous systems, the sympathetic and the parasympathetic nervous systems, that usually have antagonistic effects. Most viscera are innervated by both divisions of the autonomic nervous systems, and some authorities identify a separate part of the autonomic nervous system, that is the enteric nervous system, as a distinct autonomic nervous system subdivision. The autonomic nervous system is under the control of the brain, more specifically under the control of the hypothalamus. The function of the sympathetic nervous system is often summarized by fight, flight, or fright. Its role is concerned with preparing the body for stressful situations, increasing the cardiac output by increasing the heart rate, stroke volume, and myocardial contractility, vasoconstriction, venoconstriction, mobilizing glucose stores, and pupillary dilation. The function of the parasympathetic nervous system is often summarized by rest and digest. It carries out the basic functions required for life, including decreasing the heart rate, salivation, stimulating peristalsis in the gut, urination, and pupillary constriction. The sympathetic and the parasympathetic nervous systems often have antagonistic actions on different viscerae, but there are some exceptions to it. For example, on the heart, the sympathetic nervous system increases the heart rate and the contractility, while the parasympathetic nervous system only reduces the heart rate and has very little effect on the contractility of the heart. In the lungs, the sympathetic nervous system promotes bronchodilation, while the parasympathetic nervous system promotes bronchoconstriction and increases the production of mucus. On the pupils, the sympathetic promotes dilation, while the parasympathetic promotes pupillary constriction. On the salivary glands, the sympathetic inhibits salivation, while the parasympathetic stimulates salivation. On the arterioles, the sympathetic promotes vasoconstriction, while the parasympathetic nervous system has almost no effect on the arterioles. There is one exception to this, and is the arterioles of the penis and clitoris that have important parasympathetic innervation. On the sweat glands, the sympathetic activates sweating, while the parasympathetic has no effect. On the adrenal gland, the sympathetic nervous system promotes the release of adrenaline or epinephrine into the blood, while the parasympathetic has no effect. On the gastrointestinal tract, the sympathetic nervous system inhibits peristalsis and inhibits secretions, while the parasympathetic stimulates peristalsis and secretions. On the bladder, the sympathetic nervous system promotes relaxation, while the parasympathetic contracts the bladder. On the male genitalia, the sympathetic nervous system promotes ejaculation, while the parasympathetic nervous system promotes erection. Now we are going to see important anatomical differences of the branches of the autonomic nervous system in more detail. The action potentials that travel from the central nervous system to the peripheral viscerae travel through two sets of neurons, the preganglionic and the postganglionic neuron. This is different from the somatic nervous system that has a single neuron that is called the lower motor neuron. Now, there are differences between the branches of the autonomic nervous system. For example, their origin from the central nervous system. Another difference is the length of the preganglionic and postganglionic neuron. Another difference is the location of the ganglia, where the preganglionic and postganglionic neurons make synapses. And another important difference is the neurotransmitters that are released from the preganglionic and the postganglionic neurons that make their effects on the different viscerae that are innervated by these neurons. Regarding the origin, the sympathetic preganglionic neurons originate from the lateral horn of the spinal cord between thoracic 1 and lumbar 2 and 3. This is why the sympathetic nervous system is described as having a thoracolumbar origin or thoracolumbar outflow. These preganglionic nerves that emerge from the spinal cord then separate to form either paravertebral or prevertebral ganglia. The paravertebral ganglia form the sympathetic chain, while there are three important prevertebral ganglia: the celiac ganglion, the superior mesenteric, and the inferior mesenteric ganglia. The sympathetic chain is often divided into four parts. The cervical supplies the head and the neck. The thoracic can be subdivided into upper thoracic that supplies the heart, the lungs, and the aorta. And also we have the lower thoracic sympathetic chain that supplies the proximal gut viscerae. Then we have the lumbar or abdominal division that supplies the distal gut viscera, and the sacral or pelvic that supplies the pelvic viscera. In the ganglia, the preganglionic neurons synapse with postganglionic neurons. The neurotransmitter at this synapse is acetylcholine that acts upon nicotinic postsynaptic membrane receptors. The postganglionic neurons leave the ganglia through the gray rami communicans and travel within the peripheral nerves to synapse with their target organ. The neurotransmitter at this synapse is noradrenaline or norepinephrine, which acts upon postganglionic adrenergic receptors. There are three major exceptions to this system that we have outlined. The adrenal medulla is directly innervated by preganglionic neurons with acetylcholine as the neurotransmitter. This is because the adrenal gland is effectively a modified sympathetic ganglion that releases its neurotransmitter directly into the blood. And for that reason, we study the adrenal medulla within the endocrine system and consider epinephrine and norepinephrine as hormones in that case. Another exception is the sweat glands that are innervated by sympathetic cholinergic neurons. That means that they release acetylcholine and acts through muscarinic receptors. Another exception is the coronary arteries that also have sympathetic innervation mediated by acetylcholine. Now, we have to say that acetylcholine there will promote the release of nitric oxide during the fight or flight response, allowing the dilation of coronary arteries. In the absence of nitric oxide, acetylcholine would produce vasoconstriction in the coronaries. Now, the parasympathetic nervous system is different. Parasympathetic neurons are carried within cranial nerves 3, 7, 9, and 10, and within spinal nerves from S2 to S4, forming the so-called craniosacral outflow of the autonomic nervous system. The axons of the preganglionic neurons are very long. They synapse in ganglia that are close to their target organs. The postganglionic neurons are relatively short. The cranial parasympathetic nerves supply viscera in the upper half of the body up to the junction of the midgut and the hindgut, just before the splenic flexure of the transverse colon. The sacral parasympathetic outflow supplies the viscera of the lower half of the body. In the ganglia, the preganglionic neurons release acetylcholine, which acts upon postsynaptic nicotinic receptors. This is exactly the same as the sympathetic nervous system ganglia. At the target organ, the postganglionic neurons release acetylcholine, which acts upon postsynaptic muscarinic receptors. Now let's talk about the subtypes of acetylcholine receptors. There are basically two types of receptors. The first type is the nicotinic acetylcholine receptor that is directly linked to an ion channel. They are classified as ionotropic receptors. Nicotinic acetylcholine receptors are found in the postsynaptic membrane of the neuromuscular junction, within autonomic ganglia, and within the brain. The receptor is referred as nicotinic because, in addition to acetylcholine, nicotine acts as an agonist. Nicotinic acetylcholine receptors in the neuromuscular junction and in the autonomic ganglia are different, and they can be stimulated with different drugs. The second type is the muscarinic acetylcholine receptor, which is classified as G-protein coupled receptor and acts through an intracellular second messenger system. They can be stimulated by the fungal alkaloid muscarine, and that's why they receive this name of muscarinic. There are 5 subtypes: M1 receptors that are commonly found in secretory glands, for example, salivary glands, and in the central nervous system; M2, which are found in the heart; M3, that are found in smooth muscle of the bronchioles and arterioles; and then we have also M4 and M5, which are found in the central nervous system. Now, what are the subtypes of adrenergic receptors or adrenoreceptors? There are 4 subtypes of adrenoreceptors. First, we have the alpha-1, which are GQ coupled receptors and are found in arteriolar smooth muscle and in the urethral sphincter. There are medications that act on these receptors, for example, the vasoconstrictors noradrenaline, metaraminol, and phenylephrine, which are adrenoreceptor agonists. Doxazosine and tamsulosin are examples of selective alpha-1 adrenoreceptors antagonists that are used to treat hypertension and benign prostatic hypertrophy. The second type is alpha-2 adrenoreceptor. These receptors are found in the pancreas, arterioles, and central nervous system. Noradrenaline and adrenaline activate alpha-2 adrenoreceptors, and their activation results in vasodilation, inhibition of insulin release from the pancreas, sedation, and analgesia. Clonidine, for example, is an alpha-2 agonist that is used as a sedative and analgesic. The third type are the beta-1 adrenoreceptors, which are found in the heart and in the kidney. They are activated by adrenaline and noradrenaline, and their activation results in a positive chronotropy and inotropy. That means increased heart rate and increased contractility. Their activation in the kidneys results in release of renin that will activate the renin-angiotensin-aldosterone system. An example of a medication is bisoprolol, which is a beta-1 adrenoreceptor antagonist that is used to reduce myocardial contractility and heart rate in patients with ischemic heart disease. And the fourth type is the beta-2 adrenoreceptor, these type of receptors are found in the smooth muscle of the bronchioles and in the uterus. Beta-2 adrenoreceptors are activated mostly by adrenaline. That means noradrenaline has very little effect on them. Their activation results in bronchodilation and uterine relaxation. A medication that can be used on these receptors is albuterol or salbutamol, which is a beta-2 agonist that is used as a bronchodilator in patients with acute asthma and as a tocolytic to reduce uterine contractions in patients with premature labor contractions.